Proteolytic enzymes are enzymes that cleave the peptide bond in proteins. So what do we mean by the peptide bond? Well, we've looked at enough of these by now, but let's just draw it for fun. And this is our peptide bond, the bond between the carbonyl carbon and this nitrogen. These enzymes break the peptide bonds that maintain the primary protein structure. Well, what's the definition of primary structure? It's the amide bonds in a protein or a peptide. These enzyme specificity depend on a hydrophobic pocket. So hydrophobic pocket, and a hydrophobic pocket is a cluster of hydrophobic amino acids brought together by the three-dimensional folding of the protein chain. So if you think about primary structure, so that's the peptide bonds, then we have secondary structure, which is an alpha helix or a beta pleated sheet. Then tertiary structure is the result of the folding in of the peptide or protein chain in on itself, and that's a result of the hydrophobic interactions from hydrophobic amino acids. Examples of hydrophobic amino acids would be alanine, valine, leucine, isoleucine. And what we're going to see is that specific peptide bonds are going to be broken or cleaved by the proteolytic enzymes. And the reason why specific peptide bonds will be broken is because the amino acid residues will have a hydrophobic interaction with the hydrophobic pocket. Chymotrypsin, for example, is an enzyme that hydrolyzes dietary proteins in the small intestine. It acts specifically at peptide bonds on the carbonyl side, or the C-terminus side, of the peptide bond. The C-terminal amino acids of the peptides released by bond cleavage are methionine, tyrosine, tryptophan, and phenylalanine. Again, the specificity of chymotrypsin depends upon the presence of a hydrophobic pocket. And a hydrophobic pocket is a cluster of hydrophobic amino acids that are brought together by the three-dimensional folding of the protein chain. If you look at the structure of tyrosine, tryptophan, and phenylalanine, they have something in common. All three of them have a flat aromatic group, and that aromatic group is going to be able to fit into that hydrophobic pocket. The methyl sulfide in methionine is hydrophobic as well. Okay, now let's say we have this tripeptide that's shown down here, and we expose it to the enzyme chymotrypsin. Well, again, chymotrypsin is going to cleave a peptide bond at the carboxylic, so at the carboxylic end of methionine, tyrosine, tryptophan, or phenylalanine. The three amino acids that make up this tripeptide are alanine, so that's not one of the four amino acid residues of interest. Then we have a glycine over here on the end. And of course, I saved phenylalanine for last because it is going to be cleaved by chymotrypsin. But again, where is it cleaved? It's cleaved at the carboxylic end. So if I look at my alanine, and you might remember that we tried writing amino acids out where we went nitrogen, carbon, carbon to represent one amino acid residue, then we had another one, right? And then we would go N, C, C like that. So this would be the first amino acid, this is the second amino acid, this is the third amino acid. So we always start with the nitrogen on the left, and we should always end up with a carbon on the right. So you can see the nitrogen, carbon in alanine, and then the nitrogen and the carbon, the alpha carbon and the carboxyl carbon in phenylalanine, and then here's my glycine. Well, where is the carboxylic end of my phenylalanine? Well, I have my nitrogen here. Here's my alpha carbon, and this is the carboxylic end, right? If I just had phenylalanine, this would be a carboxylic acid, but of course I have an amide bond here, so it's an amide. So this is the peptide bond that's going to be cleaved. The peptide bond between alanine and phenylalanine, that one's not going to be touched at all because it's at the carboxylic end of alanine, and that is not one of the four amino acids that is cleaved by the enzyme chymotrypsin. This figure shows you how the R group of an amino acid, and in this case they're using phenylalanine, fits into that hydrophobic pocket. So we know that an aromatic ring that's hydrophobic. So you can imagine that inside this pocket here in the enzyme, there's all kinds of hydrophobic R groups in here. Now they're not drawn in, but they are there. And where's the carboxyl end of my phenylalanine? Well, this is the carboxyl end here of my phenylalanine. Here's the alpha carbon. So this is the peptide that's going to be broken. And in that catalytic domain, which is shown here, now they haven't drawn in any amino acid residues, but in that catalytic domain, there's going to be a serine, and the R group on a serine is a methylene, a CH2, followed by a hydroxyl. So that hydroxyl on the serine is going to be involved in the cleavage of that peptide bond. The pancreatic serine proteases trypsin, chymotrypsin, and elastase all hydrolyze peptide bonds. As the name suggests, they are produced in the pancreas and subsequently transported to the small intestine. These enzymes are the result of divergent evolution, in which a single ancestral gene was first duplicated. Then each copy evolved individually. They have similar primary structures, similar tertiary structures, and virtually identical mechanisms of action. However, as a result of evolution, these enzymes all have different specificities. These enzymes have different pockets for the side chains of their substrates. Different keys fit different locks. Chymotrypsin cleaves peptide bonds on the carbonylside of aromatic amino acids and large hydrophobic amino acids like methionine. Trypsin cleaves peptide bonds on the carbonylside of basic amino acids, and elastase cleaves peptide bonds on the carbonylside of glycine and alanine. Again, these enzymes have different pockets for the side chains of their substrates. Different keys fit different locks. This difference manifests itself in the substrate specificity. For example, the binding pocket of trypsin is long, narrow, and negatively charged to accommodate lysine or arginine R groups. Yet, although the binding pockets have undergone divergent evolution, the catalytic sites have remained unchanged, and the mechanism of proteolytic action is the same for all of these serine proteases. And in each case, the mechanism involves a serine R group in that catalytic domain.